Hello, everyone. My name is Cyril Jacob Joseph, and the date today is December 5th, 2023. This is my presentation on the 2018 Science article by Dunbar Establish All, titled Gene Therapy Comes of Age. So, just to begin with, this article centers on gene therapy. What is gene therapy? Well, it is a specific type of therapy that focuses on treating inherited human diseases using advanced understanding of genetics. The authors provide a detailed summary of the journey of gene therapy. It focuses on gene delivery vectors, such as vectroviruses and adeno-associated viruses, and also highlights the development of various gene therapy strategies, including direct in vivo administration of viral vectors. Adopting transfer of genetically engineered T cells and emerging gene editing technologies provides great promise. So, on to the evidence and the conclusion brought about by this paper. Beginning with the development of gene delivery vectors in the 1990s, initial clinical trials using gene therapy vectors like retroviruses exposed serious toxicities, including inflammatory responses and malignancies due to inserted activation of proto-oncogenes. Unfortunately, this was a setback, but the setback led to more intensified research. And then this article highlights how this setback led to the creation of safer vectors, offering gene transfer to non-dividing cells, which was seen to be more safer and effective. And it was this research that was used for eventual immunodeficiency therapies. Moreover, they also have developed self-inactivating lentiviral and beta-retroviral vectors, which also reduce the toxicity. And subsequently, the FDA began to have approval for these gene therapies, starting in 2017 for treating B-cell malignancies and congenital blindness, which is incredible. T cells are, being engineered to express specific chimeric antigen receptors that showed anti-tumor activity. And furthermore, the article also documented how we saw the rise of precise editing tools, namely with the use of CRISPR. We all love CRISPR. And while it's in its early stages, CRISPR shows promising results with regard to clinical trials. And furthermore, this article also highlights the ethical considerations surrounding germline editing. And this article talks about the heritable nature of these changes, which could have broader ethical implications. And now on to my personal opinion of this article. Well, I think this article really highlights a key factor of what the essence of genetics plays in terms of the healthcare and the clinical setting. If most disease can be treated, directed back to problems with the gene code, whether it's mutagenesis or UV damage, or chronic inflammation as a result of damage to DNA. This can have very broad implications in terms of what diseases can be treated. And if you can correct the DNA in the beginning stages, well, you could have, massive effects. And if we use the traditional model of DNA to RNA to protein, well, you could treat a wide swath of diseases from this. So I think this presents a promising delivery of the future of what genetics has for the healthcare field. And I think this article sums it up nicely, going from the very beginning and then, in the 90s with the actual clinical trials to now with the ongoing clinical trials with CRISPR. And I think it does raise up a promising ethical consideration because we're now getting into that kind of future that may have been echoed in those dystopian future books back in the past where we're editing humans and such. But I think, people can have a reasonable conversation. Guidelines can be drawn up so that there are ethical considerations made. But ultimately, it's the health of the patients that must be looked out for when we implement such genetic testing and such genetic editing. But I think this holds a great promise, and thank you for watching this presentation.